Hello, everyone, and welcome to another Applying Ethology webinar. I am Laura Whalen, a PhD student at the University of British Columbia. As we begin today, I want to remind you all to please turn off your camera and your microphone. Should you have questions for our speaker, please type them in the chat box and we will get to them at the end of the presentation. It is my pleasure to introduce Vincent Bombail, who has been a research scientist at INRAE and will soon be in Scotland with the SRUC. He will be sharing his research addressing individual differences among rats and their response to playful handling. Welcome, Vincent. You're on mute.
Okay, is it better now? Yeah, I'm assuming it's better. Thank you. Sorry. Yeah, so I'm on a sabbatical from INRAE as of a few days ago. Thank you for the opportunity to present our work today. This talk is based on an opinion paper written together with a group of us from France and the UK, listed on the screen. You can see some of their Twitter handles as well if you want to get in touch with them. I'd like to thank our colleague Taylor Hammond for some of the beautiful drawings that are shown throughout the talk. So we're talking about playful handling, which some of you will have heard of and know as rat tickling, which is a form of heterospecific social enrichment, where the human hand playfully interacts with a laboratory rat. I will not tell you right now about why I chose to use this term of a tickling in this presentation, some of this presentation. However, I hope that over the course of the talk, it becomes clear why I'm trying to make such a distinction. But this is much more of a mouthful to pronounce than tickling, so I might mix them up, and it's far easier to be selective in writing. So I will talk about individual differences in this response to playful handling, and conclude by telling you a bit more about where we want to take this research. Here, I'm trying to address a question that's relevant to you if you want to use a positive intervention to enhance laboratory animal welfare, well, rats, but also if you want to use a tool to investigate the neurobiology of positive affect, in a similar fashion as people have been using stress paradigms to look at the neurobiology of negative affect. I will start by presenting some background information about three different but interlinked subjects. First, I will talk about play behavior in rats. Then I will introduce the concept of ultrasonic vocalizations, or USVs, focusing on the 50 kilohertz range of USVs. And I will finish the introduction by showing you some examples of different methods used to tickle rats or playfully handle young rats. So if you go down to your animal facility, you'll notice that very often young rats, when housed together or socially, play with each other. You see this in cages of males and females. It's a widespread behavior that usually peaks around 30 to 40 days of age, just before puberty, and gradually declines as the rats mature. Is often referred to as rough and tumble play because of this energetic interaction. Looks like they are play fighting. Play between two rats is quite a complex affair. There are many components which I'm listing here. You can see the animals run, jump, wrestle, and you can see, like on the illustration on the screen, you can see sometimes some pinning, with one rat on top pinning the other one down. One of the first people to study play in rats was the late Jaak Panksepp, who 40 years ago published a study entitled The Ontogeny of Play in Rats, looking at how this behavior takes place in the early days, in the early developmental stages of the rats. And he came to realize that it was quite difficult to study this behavior, especially to quantify it because it's so complex and variable. He then found that if you focused on one of the play components, namely pinning, this behavior was easy to recognize. You've seen it just on the illustration. It's easy to quantify because you could just count the number of pins during a bout of play, and this correlated quite well with the amount of play in general that he observed. So Panksepp suggested that amount of play was equivalent to the number of pins observed when two rats engaged in rough and tumble play. I will return to this a bit later in the talk. Now I'll turn your attention to USV's ultrasonic vocalizations. So as humans, this isn't accessible to us. We need technology for this, but rats naturally emit ultrasonic vocalizations, and they can also hear them because we see them, they can react to them. We know they hear those sounds. This is an audiogram or a spectrogram. That's what we use in the lab to analyze the rats we record, the USVs we record from the rats. So you can visualize, so there's on the horizontal axis, you have time, and on the vertical axis, you have the frequency of the signals at any moment. And there's a color scale, so the blue color tends to be very weak signal, and when it's pink, darker, you can see it's a stronger signal. You can see the time is pretty short, so the time interval that's on the x-axis is well under 1 second, and on the y-axis you can see the frequencies at the bottom within the human auditive range, all the sounds you can hear between about 20 hertz to under 20 kilohertz, and you see the scale goes up to 100 kilohertz, which is very, very high pitch. It's an ultrasonic sound. If you were able to transform those signals, so there's an example of signals, 5 events here. You can see they're located sort of around the 40 kilohertz mark. So 5 USVs circled in red, and you can see at the bottom around the human auditive range, that's the noise that you'll get in any facility, unless you use specific special insulation from the noise. You will pick up environmental noises. So 5 USV events here, yes, and if you were to electronically modify those signals to make them audible to the human ear, they'd sound like squeaks or bird songs. So those sounds are in what we call the 50 kilohertz type of USVs. There are others, and I won't talk about them today. As I said, the time scale on this graph is pretty short, so they're very rapid events. So these are the 50 kilohertz USVs. Why am I talking about this? Well, they've been linked to positive emotions in rats. Specifically, there's been a lot of mechanistic studies, neuropharmacological studies linking activations of regions associated with reward processing, such as the striatum. So there's association drug studies showing that USV production is associated with activation of dopamine transmission in the nucleus accumbens. But for what interests us, those USVs are also emitted in the anticipation of social play, but also during social play. They're produced during playful handling and tickling, and we actually have been able to record them when they eat as well, and so we have some work prepared here. Some people would argue that they're homologous, similar to human laughter. Well, I believe it's far more complicated than this, and it's debatable. I think it's as a first approach, it's perhaps a good analogy. I'll show you data on USVs a bit later. I will now introduce the concept of tickling. Most of you will have heard of it. Perhaps you have tried it in your rats in the lab. Around the turn of the millennium, the first articles about rat tickling were published. You may recognize the name of Jaak Panksepp among the authors, the one who quantified play by counting pins. The very first article was entitled "Laughing Rats, playful tickling arouses high-frequency ultrasonic chirping in young rodents. And in this study, the authors compared different tickling methods, and they found that a full-body tickling with repeated pinning gave rise to the most USVs in the 50 kilohertz range. The tickling was described as vigorous whole-body playful tickling, focusing on the ribs and ventral surface, with animal being repeatedly pinned 4 to 6 times. Yep. 4 to 6 times throughout the 15-second interval. So yeah, that's the video now. You can find it on YouTube. You'll see there's a brief moment of tickling on the rat's neck before it's being flipped over and vigorously tickled on its belly. I'll play again. The black instrument you can see on top allows the translation of the ultrasonic sounds the rat makes into audible sounds for the humans. Usually it's a bat detector. You can also use other devices for recording. I'll show you later. I've used a drawing from Taylor that shows similarities between the pinning during social play and the pinning during the tickling, with a phase of playful biting, playful interaction with the neck, followed by the actual pinning with a rat, one of them in the case of play being flipped on its back. So it appears that pinning can be placed at a center of all the 3 subjects I've touched upon so far. It is one of the many components of rough and tumble play. It's used when rats are being tickled by a human, and it gives rise to the emission of 50 kilohertz USVs, the positive emotion vocalization. So where am I going with this? Why am I telling you all this? Questions, or maybe even problems, arise when we start to look at the responses of the rats to this type of tickling, especially the difference between individuals. This is something that would appear obvious to you as soon as you start tickling or playing with rats in the lab. They subjectively, so as an experimenter and anyone who does it really, they respond differently to the human hand as a play partner. This graph is from a recent paper from Hinchcliffe et al. They used a form of tickling you just saw an example of. On the horizontal x-axis is the rate of tickling, so the production of those 50 kilohertz USVs per minute. How much are they vocalizing? And on the y-axis is the result from an independent test of an independent measure, the effective bias test, which essentially is a measure based on choice of the animal for substrates associated with different experiences of different valence. And for the purpose of this talk, to keep it simple, it's an independent measure of how much the rats like to be tickled. The first thing we can see is that there appears to be a good correlation between 50 kilohertz USVs on the x-axis and the ABT results on the y-axis. But what's also clear is that not all rats, you can see on the circle, well, you can see that not all rats appear to be enjoying the tickling. In fact, some of these rats emit very few or even no USVs in response to the tickling. This has been shown in other studies. In fact, this has been exploited by some labs in order to investigate the biology of those different endophenotypes, we can call them, subpopulations in the rat population. Animals that respond very low to the tickling and animals that respond a lot. Now, there have been genetic studies showing that there's indeed a genetic component to this. For instance, people have used selective breeding. Panksepp did it, others did it. So that partly explains this variability, but you could also ask whether this has something to do with the method. Now, let's just have another look at this Panksepp method here. 4 to 6 pinning per 15 seconds as part of a vigorous whole body playful tickling. it says mostly playful tickling, but it's mostly pinning. So I think we can argue that this method does not contain a lot of variability and flexibility, sorry, and is fairly it doesn't have the diversity of the behaviors we've seen in the play earlier, and both are key ingredients of play. So why would it be so rigid? Well, first of all, we're scientists and we like to standardize treatments. If you work in pharmacology or toxicology, you will give each rat exactly the same dose of compound you wish to study. Actually, there's a bit of a philosophical question here. You know, you give the same dose of drug across a group of animals, but if you're studying play, which has an unpredictable component, how can we standardize and plan this as part of an experiment? But that's another question. Another point is that playful handling is difficult because exactly of this the need for the flexible response to adapt to the behavior of the rat, and the diversity of response that's also required in order to incorporate more aspects of play. I've just put the list back to remind you of the many different components observed when rats engage in rough and tumble play. So what might this playful handling method look like? There's another video. Yes, it shows you an example of a rat being playfully handled. You can see on the top left, there's a microphone, the one that we can use to record the USVs, and that allows us to visualize them with the audiograms. This animal has been habituated to the arena several times in order to remove any possible stress associated with the novelty of the new enclosure. And there, we do this by bouts of 20 seconds, interspersed with 20-second pauses in order to be able to study various aspects of the interaction. And you can see that the handler is trying to adapt his movement to that of the rat, making the hand a play partner. And also what's quite nice to see and fun to do is the rat also, you can see it running after the hand, enjoying the interaction. I can't get out of the video. Oh, yeah. So we're back to the data from the Hinchcliffe study from before using the Pankset method. And I will now introduce the results from one of our own studies. So same thing, each rat is a circle on the graph. You can see this time the rate of USV production is on the vertical axis. And this time, even though it says tickling, they were playfully handled. Half of the rats were playfully handled, and there was a control group where, in order to keep things the same, the control rats were brought to the playing arena, and the human hand was there. It just remained motionless for the time of the interaction. Well, there was no interaction. We can see that among the tickle rats, there's still a large individual variation. This is on the first day they were tickled. And we can see they're emitting more 50 kilohertz USVs than the control rats. Now, the control rats are emitting some USVs, possibly because being moved into the larger arena appears to be something more enjoyable, a break from the routine of living in a cage in the lab, perhaps, but possibly also because USVs might mean things other than simply laughter, but that's again another debate. But I think generally we can see from the responses that control animals in those studies actually find the being controls quite enjoyable, not as much as the playfully handled ones. Now I'm adding data from the fifth session. So they were tickled, they were played with 5 times, and we're showing you the rate of USV production for the animals. So it's interesting to know that all animals are producing USVs. You can see the playfully handled rats have considerably augmented the USV production. So have the controls. And you can finally see that if you look at the rate of USV production, or either the average rate of USV production in this study, is considerably higher than in the Hinchcliffe study. Now, this kind of comparison is to be taken with a pinch of salt because they were done on a different rat strain, ages differed, a variety of environmental and genetic causes as well that might explain this. Nonetheless, we had a far higher rate of USV production. So let's return to the pinning. Why might the heavy use of pinning during tickling, why might it constitute a problem? Well, repeated pinning does not resemble play, because play is much less structured and much more unpredictable. It is difficult to assess the effective state of the rat during tickling when repeated pinning is used. You can't really tell whether the rat likes it. Now, when using this method, you can't really adapt the tickling to the behavior of the rat. It's interesting to note that during social play, pinning is initiated as an avoidance response by the pin rat. The rat voluntarily rolls onto its back to avoid the more dominant rat. And the more dominant rat is doing the pinning 70% of the time. And it is also the pinning rat, the one on top, that emits the most USVs. And rats selected for low USV emission display more pins, indicating an inverse relationship between pinning and USVs. Also, much more USVs are emitted during wrestling and chasing than during pinning when the two rats play. So one question we're asking is whether USVs could be forced when we use pinning during tickling, in the same way that as humans, we can relate to the experience of being tickled but not enjoying it, and yet laughing when being tickled forcefully. Now, on this last point, there's the difference between touch and enjoyment and fun. Some studies demonstrate that during the tickling, they demonstrate the implication of the brain's somatosensory cortex areas. So they tickle rats and realize that the brain cortex regions, where the tickled regions project to, obviously they receive electric signals that then, and they found that those regions were involved in USV production, and the simple stimulation of the somatosensory cortex of these rats, so not the touch, but just activation of the brain centers involved in the touch, led to USV production, thereby implicating the sense of touch in the response to tickling. So in those cases, the touch did it, not just the fun. Anyway, when pinning is used extensively during tickling, the supine position is not chosen by the rat, and pinning by a human hand is forced rather than offered. So I will summarize and conclude on some of the points I've brought up here. Rats differ in their response to tickling. Some like it more than others. Do current tickling methods overemphasize the use of pinning? We do think so, yes. And we suggest that methods used for playful handling need to incorporate more aspects of play, so have increased diversity, be playful in other words. You can read articles by Megan LaFollette, who works in the North American 3-hour now, who has written about being playful and boisterous in her description of the tickling protocol. So increase this diversity, and also be adaptable to individual rat responses, so have increased flexibility. Again, you have a population of lab rats, they all look the same, they're all fairly genetically related, and they've been bred under similar conditions, and yet you will find quite a high degree of variability in any response you measure: novelty, reward processing, stress, social interactions, etc. So there is this element of variability. And we think that all this, oh, sorry, I forgot that. All this would lead to more positive emotions and increased welfare across your cohort of rats. So all these thoughts are described in an open access article we published earlier this year. It's available online if you're interested. I will use this opportunity to mention that some of the original thoughts on this came from a discussion. So the first time I went down to the animal facility in Paris and spoke to my colleague there, and asked if I could use 2 rats for some tickling and explain what it was, and instantly, well, it just raised concern about the possible submissive nature of being pinned for an individual. Now, this led to conversations, and I think it's a good example of how involving everyone and listening to everyone in the process can deepen the understanding of the animals we study. Now, I mentioned a game changer. So opinions can be interesting, but as scientists, we value facts. So we've recently received funding to confront our theories to the experimental reality of the laboratory. This project will be funded by the National Center for the Replacement, Refinement and Reduction of Animals in Research, NC3R, and that's a collaboration between a number of British institutions. So the plan is to compare the standard tickling technique with a playful handling protocol to mimic the dynamic nature of play I mentioned. And in practice, it's fairly simple. The aim is to test different games. So this game of tickling, just change the rules and essentially create a dose response of pinning versus effective response. And we will use this to investigate the rat enjoyment, but we'll also use it to assess whether the tickling methods, an optimized, refined tickling methods, can increase the reproducibility of research by reducing the variation of behavioral and physiological responses in the animal. Now, I'm not saying we shouldn't have any variability in animal experimentation. I mean, the process thrives on variability. I'm just talking about any sort of we'll call it stress, or any allostatic load that might be generated through issues with housing or social context, etc. And these have an impact on the quality of the data we produce through animal research. So whenever possible, we should what we do, whenever possible, we should aim should aim to benefit society. So we aim to interact with stakeholders in the UK and North America to disseminate understanding of the benefits of tickling, to encourage its wider uptake in practice. As an experimenter, I know the deal. I've been on all sides of the debate, and there's so many reasons. There's always so many reasons why our experiments might not work or might give us some difficult results to interpret. So we like routine. We like to not change things because So change is always a funny one. The other thing would be to gather data on the wider use of tickling in effective neuroscience research and as a form of enrichment. And finally, not just because it's in accordance to the wishes of our funders, but also because it's good practice, we will carry out a pre-registration of our study, and we'll set up a website to disseminate our data for outreach, really, to talk about rat tickling and playful handling, and generally about positive animal welfare for laboratory animals. Okay, well, this is it was fairly short, so I thank you for your attention, and I hope I'll be able to answer any question if you have any. Thank you.
Thank you, Vincent, and just welcoming everybody to use the chat if you have questions. For me, more of an applied question. It seems like the bigger goal, of course, is to encourage labs to actually use playful handling and tickling for the rats. And so how would you imagine a rat playpen? Would it be one where you bring several different rats together and have somebody playing with multiple ones at the same time? Like, do you have a vision for what that could look like?
So what I've showed you is because we have been using tickling, as a tool to look at learning, for instance, or essentially effective neuroscience. Now, if we were to recommend it as an enrichment, people have been working on this far longer and far better than me. I recommend some of the labs of what's the name, Brian Agaskill and Sylvie Cloutier and Megan Lafollette they've described a lot of setups really to do this, to make it more, to spread the word really. And I think I remember variations where people could do it quickly in the same cage, okay, maybe even in the home cage. I can't remember if it was a different cage, but two rats together to so that's a possibility. I mean, you could, then there are the practicalities of having many rats in a large arena. You don't want to mix up your different so I think really that there's a lot of flexibility. It depends on what people want to do and what constraints they have in the lab and in the projects. But really, I think again, it's and I'm not well, I believe the positive out of this is to essentially to ease the human animal interaction, to make something that can be, I've always found it a little bit paradoxical that we sometimes study anxiety or stress in those creatures, but the process in itself is a little bit stressful. So if we add this interaction, hopefully we'll be looking at the neurobiology or the physiology or whatever of an animal that's in a slightly more, I'll call it basal state rather than a stress-induced response.
Yeah, I couldn't agree more. Lindsay says, when engaging in play behavior with conspecifics, do more dominant versus submissive rats seek out play differently? And do submissive rats have the same drive to engage in play behavior?
So I wish I could develop more, but yes, I think there are differences in terms of submissive and less submissive individuals in how they engage in interaction. But I think I would probably say something wrong if I said more. But there's, again, a lot of people have done it and described it much better than me.
Francisca says some variations of Panskep method include hand chasing during the 15-second rounds in which tickling is not applied. Do you think this could be a form of tickle enrichment more similar to rat-rat play?
Yeah, I think exactly. I mean, I've simplified enormously, and it was almost, it's almost an extreme. The protocol is 4 to 6 bites per 15 seconds of pinning. So that's obviously there are other variations. Other protocols have been suggested, including, yes, with chasing, etc. Again, this is in the spirit of what we're suggesting. We're saying bring on that diversity, because there will be, well, back to the earlier question about social capacity, being where you stand in the group, etc. For some individuals, it might be unbearable to be tickled. So yeah, but make it a game rather than tactile, in your rat interaction.
Kind of on that theme, do you think that there's more ways to encourage rats to be, I guess, rat play besides just using your hands and tickling them and pinning them? Do you think that there's other behaviors we could do that would also encourage play? I guess thinking broadly, what are next steps for encouraging different types of rat play?
So I think, I mean, well, this one I'm speculating a little bit, but I would assume that some living arrangements might be more conducive to the development of play behavior. So maybe it's a consequence of a quality of life. I mean, it is documented that the general affective state of the animal will impact the amount of play. But interestingly, there's so Michael Brecht's lab in Berlin, they published a study where they were playing hide and seek, I think, with the experimenters. And I mean, I can't describe it in too much detail, but yeah, they looked at affective responses, like, you know, the positive emotions in response to the experimenter playing hide and seek with them, and they went into quite a lot of detail. And so there are different games that can be played. I think the rat tickling, playful handling thing is something you can do. I think, I don't even know if that's the case, but I'm assuming it's been brought on as a measure, because it's, you know, not every day, but well, I guess it depends on the lab, but very regularly the cage is put on a table, the lid open, and then something checked or something replaced, and then that could be the 15 seconds per animal. Or I mean, then again, Megan Laffollette worked out a study, published a study looking at how long was the minimum amount of time that you need to tickle your rat daily to look at to lead to enhanced affective state. Yeah, I think that the tickling story is because it's people put their hands in cages for all sorts of reasons. So why not make it a positive event rather than, you know, organize a big hide and seek in the lab, which could lead to all kinds of issues, especially if you can't find your rat afterwards.
Thank you very much, Vincent. I don't see any more questions. We can keep the Zoom open for a minute more if people are still typing away or want to ask a question. But thank you so much for your time today. I learned a lot, and it's always nice to see rats getting individual attention.
Yeah, thank you. Well, thank you for the invitation.